Okay, let's unpack this. Welcome back to the deep dive.
Good to be back.
We are about to plunge into some really intriguing and honestly a little unsettling source material. It was actually brought to us by you, our listener.
Oh, fantastic.
Yeah, this deep dive is fueled by a fascinating source shared with us, and we really want to give a huge thank you to iceni for bringing this material to our attention
Absolutely.
and, allowing us all to explore it together.
Yes, it's a perfect example of how bringing different pieces of information to the table can spark entirely new lines of inquiry. Helps us understand complex topics from, well, sometimes unexpected angles.
Totally.
So thank you, iceni. This is exactly the kind of material that makes doing these deep dives so worthwhile.
Couldn't agree more. So what fascinating rabbit hole are we jumping into today?
Well, the focus is on an audio documentary, or maybe it was a live stream discussion.
Yeah, the source calls it a live stream,
Yep.
and it poses a genuinely disruptive question. I mean, the title alone is pretty dramatic.
What is it?
Hemoglobin causes lightning-fast clot formation. With a question mark, but
Yeah.
still.
Okay, just hearing that title immediately tells you this isn't a standard textbook stuff, is it?
Not at all. It sounds like it's really challenging conventional understanding of, blood biology.
Exactly. So the mission for this deep dive needs to be clear.
Right. We're here to unpack the core concepts presented in this specific source. That's key.
Just this source.
Exactly.
Mm.
We want to understand the theory it puts forward about hemoglobin's role, the mechanism it proposes for this really, really rapid clotting.
The lightning fast bit.
Yeah, that bit.
Yeah.
And the connections it tries to draw to different health events and the pretty significant implications it discusses. The source itself even calls them chilling.
Chilling. Okay.
So our goal isn't to say if the theory is right or wrong.
No validation or refutation here.
Correct. We're simply here to deeply explore and understand the hypothesis as it's presented in this material. We're just following the narrative thread laid out by the source itself.
Think of it like we're examining a really compelling argument. We're laying it out piece by piece to see the full picture the source is trying to paint.
Exactly. And where does that picture start?
It starts by sort of redefining a molecule we all think we know pretty well, hemoglobin.
Right. Okay, so section 1 here dives straight into this. The source seems to title it something like hemoglobin, from lifesaver to potential predator.
Okay, that's quite a title.
Yeah. It begins by reminding us of hemoglobin's primary job, the role everyone recognizes.
Which is, it's fundamental to life, isn't it? The source grounds us in the known first. It describes hemoglobin's day job, its absolutely crucial function inside our bodies.
And what is that standard life-sustaining function? Just to set the baseline.
Well, normally hemoglobin is found, exclusively inside the red blood cells, those tiny sort of disc-shaped cells that make up a huge part of our blood.
The ones that make blood red.
Exactly. And the red blood cell membrane, it acts like a protective little container, right? Keeps the hemoglobin safely housed inside. And inside that environment, hemoglobin does its vital task.
Right.
It grabs onto oxygen molecules efficiently as blood flows through the lungs,
Right.
transports that oxygen all through the circulatory system, and then releases it to the tissues and organs that need it, to fuel muscles, power the brain.
Everything.
Pretty much every metabolic process needs that oxygen delivery system. Hemoglobin is that system, essentially. It's the molecule that makes oxygenated blood bright red. It's indispensable.
And the source acknowledges this.
Oh, yeah. It captures this by calling hemoglobin a dutiful molecule when it's doing its job correctly inside the red blood cell. It's part of the team doing its designated role in its proper place.
Okay, so that's the normal picture. The good guy. Hemoglobin, the vital oxygen fairy, safely tucked away doing its job.
Correct.
But the source immediately pivots, doesn't it? It introduces the scenario where things go, fundamentally wrong.
And this is where that radical shift in the source's perspective really happens. The critical event they focus on is something called hemolysis.
Hemolysis, what's that exactly?
It's the process where red blood cells get damaged and they break open. They release their contents.
They rupture, burst.
Exactly that. And when a red blood cell undergoes hemolysis, that dutiful team player hemoglobin is suddenly just released directly into the surrounding blood plasma.
So it's free floating now.
It's no longer confined within the red blood cell membrane, and the source is really clear. This is not a benign event. Hemoglobin is described as being unleashed into the bloodstream, unbound, unprotected.
Unbound and unprotected. That sounds vulnerable almost.
Or dangerous. It's now exposed to the full, complex chemical environment of the plasma, an environment it's not normally meant to interact with, at least not in this free state.
And the language the source uses here,
Yeah.
it's incredibly stark, isn't it? A complete U-turn from the dutiful molecule image.
It really is. The source does not mince words. It dramatically reframes hemoglobin's role once it's outside the red cell.
How does it put it?
The source explicitly states, and this is a line that really makes you pause, if hemoglobin escapes the red cell, it's no longer part of the team. It becomes a predator in the bloodstream.
Wow, a predator.
Yeah. It shifts it from just a helpful cargo carrier to something actively dangerous. It suggests its properties or how it interacts with things change in a way that's actually harmful to the body.
A predator in the bloodstream. That's a powerful, almost terrifying image. It completely flips the script on what we usually think hemoglobin is.
It does.
So this sets up the central mystery, right? The core question this source is grappling with.
Which is,
How could this free-floating, unbound, potentially predatory hemoglobin possibly be involved in deadly clot formation?
right. And not just any clot formation.
No, the lightning-fast kind mentioned in the title. How does that happen?
Precisely. Because, the body's natural clotting mechanisms, they involve a complex cascade of proteins activating platelets. It's a process that typically takes some time to get going and build up.
Yeah, it's not usually instant.
So how could a molecule primarily known for carrying oxygen suddenly trigger clotting at speeds described as lightning fast? It really challenges our fundamental understanding of coagulation biology.
Okay, and that leads us straight into section 2 of the source's argument, where it tries to explain the how.
The mechanism.
Exactly.
Yeah.
This section is titled something like The Mechanism, Lightning-Fast Clots and Rogue Hemoglobin. This is where the source lays out its core theory.
Yes, this is the real heart of the hypothesis being presented here. The idea is that free hemoglobin isn't just inert debris floating around after a red cell breaks.
It's not just passive junk.
No. Instead, the source suggests is it actively triggers or significantly accelerates the clotting process, and at these lightning speeds,
And they stress the speed is important.
they really emphasize that this speed is a crucial distinguishing feature. They state it's far faster than traditional mechanisms like platelet activation or thrombin signaling.
So it's not just helping the existing process along.
The source suggests it might be initiating a whole distinct, much faster pathway, not just a minor player.
Wow. Okay. The speed is key then. Because if clots can form that quickly, it could maybe explain certain medical events that seem to strike out of the blue with little warning.
That seems to be the implication. And the source tries to explain why free hemoglobin might be capable of this rapid action. It talks about its fundamental nature outside the red cell.
What does it say about its nature?
It describes free hemoglobin not as just a simple protein chain floating around, but as inherently highly reactive in this unbound state.
Highly reactive.
Yeah. Inside the red blood cell, remember, it's in a very carefully controlled environment. Outside that, its chemical properties, especially its tendency to react with other molecules, they become unmasked or maybe amplified.
Reactive meaning it's like unstable.
Yeah.
Eager to jump into chemical reactions with whatever it bumps into in the blood plasma.
That's definitely the implication. Its structure, the presence of components like iron, they make it inherently capable of interacting with other molecules.
Right.
When it's safely inside the red cell, those interactions are all directed towards its oxygen-carrying job, and any potential for causing, say, oxidative damage is managed.
Okay.
But once it's released, it's free to interact with the whole spectrum of molecules in the plasma, and some of those molecules could be crucial for regulating blood flow and clotting.
And the source gets specific, doesn't it? It mentions some particular interactions it thinks are key to this potential clotting effect.
It does. It points to specific chemical interactions as the proposed basis for this rapid clot formation. 3 main ones, it seems.
Okay, what's the first one?
First, it highlights that free hemoglobin interacts with nitric oxide.
Nitric oxide. I know that's important for blood vessels.
Exactly. Nitric oxide, or NO, is a vital molecule. It's produced by the lining of our blood vessels, the endothelium. It's a potent vasodilator, meaning it helps blood vessels relax and widen.
Which is good for blood flow.
It's essential for healthy blood flow. But critically, NO also acts as a natural anticoagulant. It inhibits platelets from activating and clumping together, basically preventing clots from forming when they shouldn't.
Ah, okay, so it's like a natural brake on clotting.
A very important one.
So if free hemoglobin is interacting with nitric oxide, is the source saying it's getting rid of this brake, neutralizing it somehow?
That appears to be the implied mechanism based on what the source presents. If free hemoglobin rapidly consumes or inactivates nitric oxide,
Scavenges it, maybe.
yeah, scavenges it, binds it up, then it would remove a crucial natural anti-clotting signal from the local environment.
And they tip to balance.
Suddenly, the balance in the blood could shift dramatically towards clotting, potentially triggering rapid aggregation of platelets or other factors.
Okay, that makes a kind of scary chemical sense. Removing a natural stop signal would definitely promote clotting. What's the second interaction the source mentions?
The source states that free hemoglobin binds to oxygen. Now, obviously, binding oxygen is its main job, right?
Right, that's what it's supposed to do.
But doing it outside the controlled environment of the red blood cell? Well, the source implies this might lead to different chemical consequences.
So,
Oxygen itself is highly reactive. And the process of hemoglobin binding and releasing it involves changes in the hemoglobin molecule's oxidation state, particularly its iron center.
okay.
The source seems to imply that this process happening out in the free plasma might contribute to oxidative stress, or maybe cause changes in the hemoglobin molecule's shape, its conformation, that somehow trigger or participate in clotting pathways.
So it could damage other molecules or maybe cell surfaces?
Potentially, or the altered hemoglobin itself becomes part of the problem. The idea is that even its normal function, when performed in the wrong place outside the cell, could become problematic.
The where and how of oxygen binding become critical then.
That's the theory being presented. And the third interaction the source flags is that free hemoglobin binds to iron.
Well, it contains iron, right? That's central to how it works.
Exactly. Hemoglobin contains those heme groups, each with an iron atom right at its center. That's where the oxygen actually binds.
Okay.
Now, free iron, or iron that isn't safely locked away within proteins like hemoglobin or transferrin, it's notoriously reactive. It can catalyze reactions that produce harmful free radicals.
Leading to oxidative damage, inflammation.
Precisely. And both oxidative stress and inflammation are known contributors to blood clotting. They create a prothrombotic environment.
So is the source suggesting that this free hemoglobin might, improperly release its iron? Or maybe the iron centers becomes exposed or reactive in a way that kicks off these damaging chemical reactions?
That interpretation definitely aligns with the source's emphasis on hemoglobin's reactive nature and its link to iron. It suggests that the iron component, normally the workhorse for oxygen transport,
It becomes a liability.
exactly, it becomes a liability when the molecule is outside its cellular home, potentially initiating or exacerbating proclotting conditions through these oxidative mechanisms.
Okay, so those three interactions, messing with nitric oxide, the problematic oxygen binding outside the cell, and the reactive iron component, these are presented by the source as the key chemical reasons why free hemoglobin might act as this rapid clot inducer.
Yes, that seems to be the core of the proposed chemical mechanism.
This paints a picture of free hemoglobin as, well, far more than just a stray molecule. And the source uses a pretty powerful analogy to capture this potential for harm, doesn't it?
It does. It uses that really dramatic description saying that free hemoglobin in the bloodstream could behave more like a chemical weapon than a helper.
A chemical weapon? Wow.
Yeah, that phrasing is clearly meant to emphasize the idea that this molecule, normally essential for life, becomes actively toxic and damaging when it's displaced.
So it's not just passive, it's chemically aggressive.
That's the idea. Interfering with vital processes like nitric oxide signaling, maybe mishandling oxygen and iron outside the red cell, it's depicted as an internal threat that's been unleashed.
It really drives home the potential severity of this mechanism if the source's theory holds any water. And the source also describes the nature of the clots themselves. Are they supposed to be different from typical blood clots?
According to the source, yes, potentially quite different. They suggest that clots formed by this free hemoglobin mechanism might have distinct characteristics compared to those formed mainly through the standard coagulation cascade, involving platelets and fibrin.
How are they different?
The source states these clots may be faster, harder, and more difficult to dissolve.
Okay, let's break that down. Faster to form, we covered that, the lightning speed.
Right.
Harder, what does that mean? Physically tougher.
That seems to be the implication. Perhaps more densely packed, more rigid. A harder clot might be less likely to deform and squeeze through narrow blood vessels, making a complete blockage more likely.
Okay, that sounds bad. And more difficult to dissolve.
Yeah. Resistant to the body's natural clot-busting processes, or maybe even resistant to medical interventions designed to break down clots.
So resistant to fibrinolysis, the body's own system.
That would be the implication. Fibrinolysis relies on breaking down the fibrin mesh that typically forms the structure of a clot. If these hemoglobin-driven clots have a different structure, maybe less reliant on fibrin or somehow protected from the enzymes involved,
They might just stick around longer or be harder to treat with clot-busting drugs.
that's the potential consequence highlighted by the source. Think about that combination. Forms almost instantly, potentially harder and more obstructive and resistant to breakdown.
Yeah, that combination.
Mhm.
It makes these hypothetical clots sound potentially far more dangerous, more likely to cause severe or permanent damage downstream of the blockage.
It really raises the stakes.
Okay, so we've established the core theory from the source. Free hemoglobin, let loose from red blood cells, becomes this highly reactive entity.
A chemical weapon.
Right. Interfering with nitric oxide, handling oxygen and iron in problematic ways,
Leading to lightning-fast clots.
which are potentially harder and resistant to dissolution. That's a very specific and pretty concerning mechanism being proposed.
It is, and the source doesn't stop there, does it? It then tries to connect this theoretical mechanism to things we see in the real world.
Looking for patterns, conditions where this theory might offer an explanation.
Exactly. Which brings us to Section 3 in the source's narrative, connecting the dots.
Right, where the source looks at historical data, current events, places where this free hemoglobin clotting idea might be relevant.
Yes, it casts quite a wide net here, looking at different medical conditions and scenarios that might involve significant red blood cell breakdown hemolysis, or perhaps systemic stress or inflammation that could interact with free hemoglobin.
And which specific conditions does the source mention?
The source specifically mentions connecting this theory to conditions like sickle cell anemia.
Okay, known for red blood cell issues.
Right. Also malaria.
Also involves red blood cells.
Definitely. And then it mentions more recent events like COVID-19 vaccine reactions.
That's a pretty diverse set of conditions and events. How does the source imply these are linked back to its free hemoglobin theory? What's the connecting thread, just based on the source material itself?
Well, the implied connection, just looking at what the source presents, seems to be rooted in situations where either A, significant hemolysis occurs, releasing that free hemoglobin.
Like in sickle cell or malaria.
Exactly. Or B, situations where there are maybe systemic inflammatory responses that could either potentially cause some hemolysis, make cells more fragile, or perhaps exacerbate the effects of free hemoglobin if it's already present for other reasons.
Okay, let's take sickle cell anemia as an example. How does the source seem to connect it?
Well, in sickle cell, the red blood cells have that abnormal sickle or crescent shape, especially when oxygen is low.
Right.
And those abnormally shaped cells are much more fragile. They're prone to breaking apart hemolysis as they try to squeeze through tiny blood vessels.
So there's chronic hemolysis.
Chronic and sometimes acute episodes of massive hemolysis. This releases significant amounts of hemoglobin into the plasma. The source seems to be suggesting that the well-known clotting complications, the painful vaso-occlusive crises that people with sickle cell experience, could potentially be linked at least partly to this repeated or ongoing release of free hemoglobin and the lightning-fast clotting mechanism it proposes.
Okay, so linking a known complication to this specific proposed mechanism, what about malaria?
Malaria is caused by a parasite, Plasmodium, that infects red blood cells.
Inside the cell.
Right. And as the parasite multiplies inside the red cell, it ultimately causes the cell to rupture. This releases new parasites to infect more cells, but crucially in the context of this theory.
It also releases the hemoglobin.
Exactly, into the bloodstream. Malarial infections, especially the severe forms like cerebral malaria, are characterized by significant hemolysis. The source appears to be suggesting that the severe, often life-threatening complications of malaria, things like microvascular clotting, organ damage, could be connected to this massive release of free hemoglobin during the destruction of infected red cells.
Again, linking a known outcome to this specific theory.
Yes.
And then the source also brings up COVID-19 vaccine reactions. That feels, well, much more recent and potentially controversial.
It is placed alongside these other conditions by the source, suggesting it sees a potential relevance there too.
How does it make that link?
The source doesn't seem to detail the specific pathway beyond just mentioning it, but listing it suggests the source sees a possible connection, perhaps implying scenarios where, say, inflammation or immune responses or other physiological effects triggered by the vaccine could potentially lead to some degree of red blood cell fragility or maybe enhance the reactivity of any free hemoglobin present.
Contributing to some of the observed clotting events that were sometimes associated with certain vaccines.
That seems to be the implication. It's presented as another phenomenon the source suggests might potentially fit within the explanatory power of this free hemoglobin theory. But it's more of a suggestion of a link rather than a detailed explanation of how that specific link would work based on the material we're looking at.
Okay, so the common thread the source is weaving across these diverse examples seems to be situations involving significant red blood cell damage and hemoglobin release.
Or maybe conditions involving inflammation which could interact with this mechanism.
Right. And
Um,
the source emphasizes that this potential effect isn't necessarily the same for everyone, does it? It mentions amplifying factors.
Yes, that's a key point it makes. It highlights that the impact of free hemoglobin might be significantly worse in certain individuals with underlying inflammatory conditions.
So if someone already has chronic inflammation, or maybe an acute infection causing inflammation, they might be predisposed to a much more severe reaction if free hemoglobin gets released.
That's exactly what the source suggests. Underlying inflammation could potentially make red blood cells more fragile in the first place, increasing the chance of hemolysis.
Okay.
Or, even if hemolysis happens for another reason, the inflamed environment itself could enhance the reactive nature of the free hemoglobin. Maybe make that nitric oxide depletion worse, or amplify the oxidative damage pathways.
Making the whole situation more dangerous.
Right. The source uses some stark language here, too. It warns that in individuals with these pre-existing inflammatory conditions, the consequences could strike without warning.
Strike without warning, implying a sudden, unpredictable event, triggered by this combination of free hemoglobin hitting an already inflamed system.
That's the grim picture it paints. It adds this layer of hidden vulnerability. It suggests it's not just about the presence of free hemoglobin, but also about the state of the body, the terrain it enters.
And there was one other factor the source mentioned, something about nanostructures
Hmm.
or hydrogels. That sounded quite different.
Yes, it is presented as a separate but potentially compounding factor. The source raises the possibility that nanostructures or hydrogels in our bloodstream, whether natural or synthetic, could be amplifying this reaction.
And nanostructures or hydrogels,
Yeah.
amplifying the reaction. What could the source possibly mean by that, based on the material? It sounds a bit sci-fi.
Well, the source doesn't seem to provide extensive detail on what these structures or gels are or where they might come from, natural or synthetic, it says.
Right.
But the implication is that certain microscopic structures, or perhaps gel-like substances that might be present in the bloodstream, could somehow interact with the free hemoglobin, or with the clotting process it initiates, in a way that makes things worse.
How might they do that?
Maybe they provide surfaces that help blood components stick together. Or perhaps they somehow enhance the chemical reactivity of the free hemoglobin itself. It's not fully spelled out.
So it's presented as another potential variable, an additional factor that could potentially worsen the proposed free hemoglobin clotting mechanism.
Exactly. It suggests the issue might be a complex interplay between the free hemoglobin and other elements that might be present in the blood, whether they occur naturally or are introduced somehow.
Okay, so the theory builds. Free hemoglobin gets loose from damaged red cells, becomes this reactive chemical weapon,
Interacting with NO, oxygen, iron.
causing these lightning-fast, hard-to-dissolve clots.
Right.
And the source connects this to conditions known for hemolysis or inflammation. Sickle cell, malaria, maybe even suggests a link to COVID-19 vaccine reactions.
And proposes that underlying inflammation and maybe even these other nanofactors could amplify the whole dangerous effect.
Wow, okay. That effectively summarizes the core hypothesis and its potential contributing factors, as presented in the source material we're examining.
It's quite a narrative, isn't it? Takes a molecule absolutely central to life and posits its potential to become a major internal threat under certain conditions.
It really does. And this naturally leads into what the source frames as the chilling implications. Section 4, where the source really gets into what this theory could mean for real-world health outcomes and maybe for our whole understanding of disease.
Yeah, this is where it explores the potential consequences, the broader societal implications, if this free hemoglobin clotting theory turns out to be accurate, or even just a significant contributing factor. And the source uses words like darker and chilling to frame these implications.
Suggesting they challenge existing paradigms pretty heavily.
Definitely.
And the source doesn't shy away from connecting this theory to some really serious health crises, does it? What are the specific manifestations or symptoms that the source suggests could potentially be explained by this mechanism?
It explicitly links its theory to several really concerning health events, things that have been observed, perhaps with increasing frequency, according to the source. These include
More.
the rise in sudden deaths,
Okay, that's huge.
also unexplained strokes.
Strokes without a clear cause found through normal tests.
That seems to be the implication. Also, heart attacks, and it specifically mentions vaccine-related clotting events, again here in the implications section.
So the source is basically putting forward this single underlying mechanism, free hemoglobin, causing these rapid, hard clots as a potential explanation, or at least a significant contributor, to a whole range of seemingly disparate, often sudden and severe medical emergencies.
That is the really provocative suggestion being made here. The source positions its theory as a possible unifying explanation for some of these alarming trends or events,
Instead of looking at each one in isolation.
or solely through the lens of conventional clotting mechanisms.
Right.
The source asks the audience to consider, what if a fundamental issue with free hemoglobin could be an overlooked common denominator, or at least an amplifier in these situations? It's posing a what-if scenario, but one with potentially devastating real-world consequences if true.
And that brings us to a really central part of the source's argument about these implications, the whole overlooked cause angle. If this free hemoglobin theory is valid, what does that imply about how these events are currently being diagnosed and understood, within the medical system?
This is where the source's framing becomes particularly critical, I think. It asks directly, could hospitals be overlooking the true chemical cause behind clot-related events? Think about it. If, as the source proposes, free hemoglobin triggers clotting through mechanisms that are distinct from, or maybe just much faster than, the standard coagulation cascade.
The cascade involving factors like thrombin and fibrin, or platelet activation?
Exactly, which are the focus of many of our current diagnostic tests for clotting disorders,
Right.
then it's entirely possible that conventional medical tests might simply not be detecting this underlying cause.
It's like a mechanic only checking the fuel injection system when the real problem is a faulty spark plug.
That's a very apt analogy, yeah.
They're looking at the wrong part of the engine, or maybe just not all the relevant parts, because their diagnostic tools are focused on the most common, well-understood issues.
Precisely. The source is suggesting that because medical science has traditionally focused so heavily on platelet and fibrin-based clotting, a fundamental chemical mechanism, one initiated by free hemoglobin, could be entirely missed. Missed during investigation, diagnosis, maybe even during autopsy.
And the source uses language like unlocking buried truths.
Yes, implying that a fundamental piece of the blood clotting puzzle might be hidden in plain sight, right there, within one of the blood's most essential molecules.
Hemoglobin.
Simply because we haven't been looking for it in this context, or perhaps in this state, free and unbound. It suggests our current understanding of clotting might just be incomplete, and critically, missing a potentially rapid onset, dangerous pathway.
This isn't just suggesting a minor tweak to medical understanding, is it? It sounds like a fundamental challenge.
Oh, absolutely.
And the source poses a really dramatic question about medicine's readiness to even face this possibility.
It does. It questions whether medicine is prepared to accept the possibility that hemoglobin, this molecule, universally revered as a life giver for its oxygen role,
Could also, under certain conditions.
become a possible death bringer. That's the stark term the source uses.
Wow.
It really highlights the profound paradigm shift that accepting this theory would require. It means confronting the, frankly, uncomfortable idea that a core component of our own biology, when it gets displaced, could become a source of internal pathology, capable of causing sudden catastrophic events.
What would that mean practically?
Well, it would necessitate a reevaluation of diagnostic approaches. For one, maybe the development of new tests specifically designed to measure and assess the state and quantity of free hemoglobin in the blood.
And maybe new treatments, too.
Potentially entirely new therapeutic strategies aimed at either preventing hemolysis, neutralizing free hemoglobin, or maybe counteracting its specific downstream effects, like the nitric oxide depletion.
It forces you to question something you thought was settled science, the basic role of hemoglobin.
Completely.
And the source concludes this part of its argument with a pretty strong call to action, doesn't it? Urging listeners to reconsider their assumptions.
Yes, the source's closing statement in this section is a direct challenge. It urges people to question everything we thought we knew about the blood flowing in our veins.
That's the ultimate message, really.
It seems to be.
Yeah.
If a molecule as fundamental and, seemingly well understood as hemoglobin, can harbor such a dangerous potential just by being in the wrong place. Well, what else might we be missing?
What other critical aspects of our own complex biology might be overlooked?
Exactly. It positions this free hemoglobin theory as a reason to adopt a stance of perhaps healthy skepticism towards existing knowledge and to encourage deeper investigation into these really fundamental biological processes.
Okay, so let's just quickly recap the extensive journey this source material has taken us on. It started by establishing hemoglobin's vital, life-sustaining role inside the red blood cell.
Right, the baseline. Then it introduced the critical concept of hemolysis red cells breaking open, releasing that hemoglobin into the bloodstream.
And it proposes that this free-floating, unbound hemoglobin isn't passive. It becomes a highly reactive chemical weapon,
Potentially causing damage through specific interactions messing with nitric oxide, problematic binding of oxygen and iron outside its normal cellular home.
leading, according to the source's theory, to the formation of lightning-fast clots.
Clots that are potentially harder and more difficult to dissolve than typical clots.
And the source then attempts to connect this proposed mechanism to various real-world conditions known for hemolysis or inflammation.
Like sickle cell anemia, malaria.
And it even suggests a potential link, however vaguely defined, to COVID-19 vaccine reactions.
While also suggesting that underlying inflammation and maybe even other factors like these mentioned nanostructures could amplify the dangerous effect.
Culminating in what it calls the chilling implications.
Right. The idea that this potentially overlooked mechanism of free hemoglobin clotting could be a significant factor, maybe even a primary cause, behind phenomena like sudden deaths, unexplained strokes and heart attacks, and certain vaccine-related clotting events.
And suggesting that our current medical diagnostics might be missing this true chemical cause.
And challenging medicine itself to recognize hemoglobin's potential dual role, not just life giver, but also possibly a death bringer under the wrong circumstances.
The source is definitely presenting a coherent, step-by-step hypothesis, linking a specific biological event hemolysis to a proposed rapid clotting mechanism, and then attempting to connect that theory to observable and concerning health outcomes.
It's a powerful narrative, and one that explicitly urges a reconsideration of some fundamental biological assumptions.
And the source material itself, the live stream discussion, it encourages further exploration of these ideas, doesn't it?
Yes, it concludes by telling the listener, watch the live stream, study the science.
It's basically a call for you, the listener, to delve into the material yourself and make up your own mind.
Exactly. It frames this theory not as settled fact, but as something that demands further investigation and consideration, inviting the audience to look at the evidence and reasoning presented in the original source.
So this deep dive, which was driven entirely by the source material, brought to our attention by Iceni. Thank you again for that.
Yes, thank you.
It's been focused completely on understanding the contours of this particular theory, the potential dangers of free-floating hemoglobin and its proposed role in rapid clot formation, as laid out in that source. It really does make you think about the incredibly delicate balance that exists within our own bodies.
It absolutely does. It takes something so familiar, so fundamental to how we think about blood and life, hemoglobin, and presents this scenario where it could become a source of internal breakdown, internal danger, simply by being out of place.
Yeah. So here's a thought for you, the listener, to ponder as you continue to explore the world around you and the biology within you. It builds directly on the core concepts we've explored today from this specific source material, but it's really a question for you, not an answer from us.
Okay.
If the body's own fundamental components, molecules like hemoglobin, which are absolutely essential for delivering life-sustaining oxygen, if they can become predators, as the source puts it, capable of causing sudden catastrophic damage just by being displaced from their intended cellular environment, what does that reveal about the incredible complexity and maybe the inherent precariousness of the biological systems that keep us alive every second?
Yeah.
And maybe what other critical yet currently overlooked mechanisms might be lurking, hidden in plain sight within our own physiology, just waiting for us to truly understand them?
That is a challenging perspective on the familiar, isn't it?
It really is. Well, thank you for joining us for this deep dive into the fascinating and yet somewhat unsettling hemoglobin clot mystery, as presented in the source material shared with us by Iceni.
Stay curious out there.
Stay conscious. We'll talk to you next time.